Hello, welcome back to my video tutorial for single-cell RNA and ATAC data analysis. From today, I'm going to make videos to show you how to use Monocle3 and perform single-cell trajectory analysis. So you already see my video about the RNA velocity. We can use scVelo to perform trajectory analysis, but the use of RNA velocity method is more complicated for our users because most packages were developed for Python, and also RNA velocity analysis needed NUM files, which is not publicly available for us to download it. Monocle3 is a R-based package and it is much easier to use. So probably you know we can use Monocle3 for trajectory analysis, but actually Monocle3 can perform cell clustering. You can see from the online tutorial, here is the workflow for Monocle3. It actually can start from the single-cell RNA-seq data. Later we will make videos for single ATAC-seq data. Then do the pre-processing dimensional reduction, then cluster cells, and also it has function to identify the markers to annotate the cell clusters. Then the last step is for the trajectory analysis. Because I use scRNA to analyze single-cell RNA sequencing data in R. In the beginning, I don't bother to learn this process. I just want to convert my scRNA object into Monocle3 CDS object, then perform trajectory analysis. So I tried the Monocle3, the standard process. Actually, it is very easy to use. So today I'm going to make the first video for analyzing the cell clusters. In my second video, I will show you how to perform the trajectory analysis using Monocle3 analyze the dataset. So let's have a look at the basic requirement for Monocle3 package. You can see Monocle3 store the data as cell dataset object. So later you will see in our analysis, we always named the object as CDS. Then Monocle3 has a function called align CDS to remove the batch effect. Next, we can perform dimensional reduction and cluster the cells. So let's see how to start the analysis. Here is the get started. Of course, you need to install the package. There's the instruction here to show you how to install the Monocle3. Of course, we need other packages. Depends on what kind of analysis you are going to do. So first thing to do is to load the data into R. For me, if you can load your data correctly in any analysis, actually the rest analysis is not difficult. You just need to understand the function of packages. You can actually copy code from online tutorials to perform the analysis. The online tutorial explained the structure of the data to load into R for Monocle3 analysis. So you can see here, Monocle3 store data as a cell dataset object, and when we start to load the data, it requires 3 data files to start with: the expression matrix, the cell metadata and the gene metadata. So in my tutorial, I'm going to show you how to load the data from the cell ranger pipeline. I want to show one important thing to you here is that for the gene metadata, one column should be named as gene short name. We need to do this for the feature files from cell ranger pipeline. So for today's demonstration, I'm going to use the GSE 132771 dataset again. So this is the dataset that performed the single-cell RNA-seq analysis in human fibrotic lung. And I want to analyze where the fibrotic cells come from. So down here, you can see a Monocle dataset. We are going to use the sequencing data for flow cytometry purify the mesenchymal cells from IPF lung. So they have 3 repeat IPF1. Nin represent purified mesenchymal cells. The second repeat IPF2 and the third repeat IPF3. So if you want to use my dataset for practice, you can download from here. I downloaded it already. We can have a look at the files in my computer. You can see here I have the IPF1, 2, 3 dataset. We click one and have a look. You can see inside we have the barcodes file, feature file, and the matrix file. When you download the data, they are in the compressed format. You need to unzip it. So we have the data. Now we are ready to do the performance in R. So first let's load the packages. Monocle3. I'm using the tidyverse. And also I need patchwork to show figures together. Then we set a random set. So now we can use the load data function from Monocle3 to load the first dataset from IPF1. So you can see we just tell R where my data is, the matrix data, the feature data, and the barcodes. So we load this one as CDS1. So you can see we have a large cell dataset in the environment window. So let's have a look at the column data. This is more like the metadata in thread analysis. If we run, you can see we have the barcodes in the row names. Because we are going to load 3 repeats, so we want to label the data where they come from. So we can add a column in the code data named the sample, and also for the CDS1, the data come from IPF1, so we use the paste function to label all the cells as IPF1. Let's do this. Then we can have a look at the code data again. You can see now we have a column here named as samples, and this data set for CDS1, we know all the samples are come from IPF1. Now we can do the same to know the CDS2 from IPF2, and also we label the cells as IPF2. Then we know the IPF3 as CDS3, and also label the cells as IPF3. So you can see we have 3 CDS dataset here now. We want to merge them together to do the analysis. So we can use the combine CDS function to list the 3 dataset and combine them to one as CDS. Let's do this again. So you can see we generate a CDS dataset including 3 repeatable RNA sequencing dataset. We can connect each dataset and have a look at the data structure. For example, CDS1, you can see here is the information how we store the data into the CDS dataset and also the analysis. At the moment, because we haven't do analysis, I want to show you here it's assay data. You can see we have the RNA assay. We have 33,694 genes and 4,395 cells for CDS1. If we have a look at CDS2, we have the same amount of genes and more cells in CDS2. For CDS3, you can see still the same amount of genes, and this one has nearly 13,000 cells. If we have a look at the CDS object, you can see we have the genes here, then the total cells from all 3 datasets. So we are going to do the analysis on the CDS object, then we can remove the CDS1, CDS2 and the CDS3. So the gene information was stored in the feature data of CDS. Let's have a look at the gene information. You can see here the first column is the gene ID and the second column is the gene name. You can see here the gene column was named just as V2. Remember, in the beginning I introduced we need to label this column as gene short name. If you don't name it, when you identify genes in the cells, you only get the gene ID. You cannot get the gene names. So let's change the column name to gene short names for the feature data. So we can have a look again. You can see now the column name is changed to gene short name. Now we can do the pre-processing step. In this step, it performs the data normalization and also the principal component analysis. For the online tutorial, it suggests you use a high principal component. For the initial analysis, you can check it later to see if actually you used enough PC for your analysis. But if you increase the PCs, it takes longer for the computer to run the analysis. So let's do the pre-process step, and we use the PC as 100. We can run this step. Okay, the computer finished the step of pre-processing, so we can use the plot variance explained function to see if we used enough PC to catch the most variant in gene expression across all the cells in our dataset. So let's run this function, and it is a similar function like the elbow plot in Surat. You can see it generates a figure like the elbow plot, and it shows the PCA in the x-axis. From the Surat analysis, the elbow plot, we know if we use like 25, PC should be good enough for our analysis. So let's just keep the 100 PC for downstream analysis. So next step is to perform non-linear dimensional reduction. We can use the UMAP or t-SNE. The default assay for reduced dimension function in Monocle3 is UMAP. I personally recommend you use UMAP because UMAP is better to do the trajectory analysis. The cell canisters in T3 analysis are well separated from each other. So let's run the reduce dimension function to perform the dimensional reduction. You can see down here we didn't specify which method, so the default method is PCA. So we can use PCA for the single-cell RNA sequencing data analysis. Neat, when we use the analysis for single-cell ATAC dataset, we will use the LSI function because that function is better for ATAC-seq data analysis. So we finished the dimensional reduction analysis. Now we can visualize the cells. So let's have a look at the code data again because we can use any column name here to group the cells. So you can see here we named the samples as IPF1, IPF2 and IPF3 in the samples column. So now we want to see the batch effect from the analysis. So we are going to plot the cells and count the cells by the samples, which means we are going to label the cells by IPF1, IPF2 and IPF3. So when you run the plot function, Monocle3 always asks you if we want to do the trajectory analysis. If we want to do that, we need to use the function learn graph before we do the plot. So at the moment we are doing the cell clustering, so don't worry about that ordering. So you can see here in the plot windows, IPF1 samples were labeled as red color, IPF2 are in green color, and IPF3 are in blue color. You can clearly see there are very strong batch effect between three samples, and the cells were not integrated together from three sequencing analysis. So next we need to remove the batch effect. In the Monocle3, the function called align the CDS. So this is the function that we do data integration in Surat. So we use the 100 PC for the pre-processing. So we are going to use the 100 PC to remove the batch effect, and the alignment group will be the sample. Let's run this function to remove the batch effect between three samples. You can see down here there is a publication to explain the function for remove batch effect. If you want to know the details, you can read this paper. So now we remove the batch effect. We need to run the reduce the dimension function again. Now we can plot the cells to see how the UMAP looks like after we remove the batch effect. So you can see here now all three datasets were integrated together. So that means we remove the batch effect. Now we can do the downstream analysis. So next step is to cluster the cells. This is a similar function as find the clusters in Surat. So we can also set the resolution. High resolution you will get more cell clusters, and the low resolution you get less cell clusters. So let's run the function to cluster the cells. Now we can plot the cells again to see the cell canisters. You can see here we have 8 cell canisters. So if you don't know the cell data well, then it's hard to know what kind of cells in each cell canister. So we can use the top marks function to find the markers for each canister. And this is a similar function like find the markers in Surat. Let's run the top markers function to find the markers for each cell canister. After finish the running, R will generate a file. We named it as mark tested. So you can see we have data from here. It contains 200 genes and 10 variables. Let's have a look at the data set. You can see here in the column 1 is the gene ID, second column is the gene short name, then the third column is the cell group. There are other parameters such as mark score, mean expression. We can use the column name here to arrange the markers for each cell canister. Because we have 200 markers for 8 cell canisters, it's hard to see which one is the best from here. Then we can reduce the number to just keep the top markers for each cell canister. We can filter the 200 markers by the fraction expression bigger than 0.1. Then we can group the cells by the canister and only show top 3 marker. And we ranked the marker according to their marker score for each cell canister. Let's run. You can see we generate a data frame containing 24 genes. Because we have 8 cell canisters, we only keep the top 3 genes for each canister. We can get the gene ID for the markers, then we can do the plot to see the markers for each cell canister. So let's plot the markers. So you can see we generate a plot for the 24 genes. So let's plot the marker genes together with the cell canister. Then it is easier for us to look at the markers for each cell canister. Let's plot them together. Okay, we can increase the windows. Let's plot it again. We can zoom in to have a better view. So you know we only keep 3 markers for each cell canister. So for canister 1, these 3 markers, NAM, PT, GDS, and FBL1 are the best markers for canister 1. We know NAM is a mark for fibroblast, then canister 1 is a fibroblast. So next 3 markers will be good for canister 2. You can see we have IGF-BP7, RGS16 and RGS5. So we know especially the RGS5 is a marker for parasites. So now we know cells in canister 2 are parasites. Then the markers for canister 3 is MLY9, TAGL1, ADI-RF. So they are marker genes for smooth muscle cells. Then canister 3 are smooth muscle cells. So for CNAST4, the best marked genes are CLU, MYLK, and ACG2. So they are markers for the fibroblast in the airway. So cells in CNAST4 are airway fibroblast. You can see CNAST6 express CD55, so it's a marker for immune cells, and the CNAST7 express FTPB. They are markers for the AT2 cells. And the CNAST8 express the marker KRT17. So KRT17 is a marker for basal cells in the lung. So we know from all three data sets, the cells are purified from lung as mesenchymal cells, but we still got a small amount of immune cells, epithelial cells, and the CNAST5 express UPK3B, so the cells are from the pleural tissue. They are mesothelial cells. So one possibility we have those cells is the antibody is not 100% specific to the mesenchymal cells. And you know that for experiment, this is normal. When we purify cells by flow cytometry, you cannot get 100% pure cells. So now we know the name for each cell canister. Then we can label the cell canisters. So the last step for today's demonstration, we are going to annotate the cells. According to the marked gene expression in each cell canisters, we already identified by the marked genes for each cell canisters. Canister 1 is LV and fibroblast, canister 2 is parasite, 3 is smooth muscle cells, and then airway fibroblast for canister 4, mesothelial cells for canister 5, immune cells for canister 6, AT2 cells for canister 7, and the cells in canister 8 are basal cells. So let's have a look at the canister information in the data set. You can see the canister number underneath of each cell barcode. Then we can generate a new column named as assigned cell types in the code data and store the canister information as characters. So let's run this code and we can have a look at the code data. You can see now we have a column named as assigned cell type, and we have the canister information for each cells in this column. So now we can use the recode function in the dplyr to name the cell canisters. We already know what kind of cell canisters they are from here, so we just need to name it. We can have a look at the code data again. You can see now in the column of assigned cell canisters, each cell was labeled by their cell type. So we can plot the cells and colored by the cell canister name. Let's run the plot function again. We can zoom in. You can see now the cell canisters are labeled by fibroblast, basal cells, mesothelial cells, parasite, smooth muscle cells, immune cells, and AT2 cells. So we went through the standard workflow analysis using Monocle3 to analyze single-cell RNA sequencing dataset. You can see lots of functions are very similar to SURAT, and it is very easy to use. So I recommend you can try Monocle3 to canister your cells. Then you don't need to convert your SURAT object for trajectory analysis. So that's it for today's demonstration. We can save the CDS object. The next time we just need to read it into R and perform trajectory analysis. Okay, I'm going to stop from here. I hope my video tutorials can help your data analysis. Please subscribe my channel, share my channel with your friends. I'm creating a free channel to help everyone's analysis. My reward is your subscription and positive comments. Thank you and see you in my next video for the trajectory analysis.